Now next let us see what renal corpuscle is all about. Another name for renal corpuscle is Malpighian body. It is named in honor of a famous scientist Malpighi because he has done a great deal of work in study of excretory system. So this particularly renal corpuscle part of the kidney is known as Malpighian body. Now what you have to pay attention over here is, because we have studied the structure of nephron, so we know that this is glomerulus, the tuft of capillaries is glomerulus. This is the afferent and efferent arteriole. So this happens to be afferent arteriole and this is efferent. Alright, this entire part is glomerulus. This is Bowman's capsule. Again, same labeling, same understanding, and this is PCT. So what you have to pay attention over here is that there is finger-like enfolding of the Bowman's capsule's epithelium, that is again squamous epithelium, and the epithelium of the capillaries, and it leads to the formation of podocytes. What are podocytes? Podocytes are slit-like apertures through which the ultrafiltrate enters the Bowman's capsule. So over here you are going to find podocytes, the slits which would be present, so that this filtrate which is present inside the renal corpuscle, the arteriole or the glomerulus, it would fall into the Bowman's capsule and from there it would fall into PCT. This is what takes place inside a Malpighian body, because over here this is the site of ultrafiltration. Why it is important? Because the ultrafiltration takes place inside this part of kidney where maximum of the blood is able to throw its content due to these slit-like apertures known as podocytes into the Bowman's capsule, because the capillary network that is present, it is also having a thin epithelium. There is a basement membrane in between. It's a similar way as we do it in all the epitheliums over where diffusion takes place. There is a thin epithelium, squamous epithelium of the capillary system, then there is a basement membrane, and then there is epithelium of the Bowman's capsule, and in between there are invaginations which are formed due to the tuft of capillaries, and slits are formed, that is known as podocytes. So basically this is all about the renal corpuscle, and why it is important, because it is the site of ultrafiltration that takes place inside a nephron. Now the next topic that we have to understand along with this renal corpuscle thing is that how many types of nephrons are present inside the kidney. Within a single kidney there are two types of nephrons which are present. First one would be cortical nephron and the other one would be juxtamedullary nephron. As the name suggests, these nephrons will be in maximum amount present in the cortex of the kidney. You know that kidney is divided into two parts, that is the renal capsule. Inside the renal capsule we have outer cortex and inner medulla. So the cortical part is having maximum of the nephron content. If that happens, that nephron would be known as cortical nephron. That is, again recall the structure of nephron, that the PCT, the DCT, the glomerulus, they are otherwise also present in the cortex itself, but if the loop of Henle has descended greatly into the medulla, then that nephron would be known as juxtamedullary nephron, and if it is not that much descended into the medulla, then the nephron is known as cortical nephron. That means a small amount of loop of Henle descends into the medulla of kidney. The nephron would be known as cortical nephron, which suggests that maximum part of the loop of Henle and the nephron would be present in the cortex itself. When it extends deeply into the medulla, then the name over here suggests juxtamedullary. The nephron will have a longer loop of Henle extending greatly into the medulla, that would be known as juxtamedullary nephron. Now we are going to come across another word, juxta, which would have juxtaglomerular. I hope you will not confuse yourself with juxtamedullary nephrons and juxtaglomerular apparatus. Now this apparatus is very important. We are going to study about it in the formation of urine itself. So for now what you have to remember is that there is a renal corpuscle which takes part in ultrafiltration, which is the actual site of ultrafiltration, and there are two types of nephrons, one is cortical and one is juxtamedullary. While discussing the structure of nephron, we had discussed that longer is the loop of Henle, more would be the reabsorption of water that would be taking place. So in case of juxtamedullary juxtamedullary nephrons, the loop of Henle is quite longer, and the mammals which are able to reabsorb maximum of the urine will have juxtamedullary nephrons. So, I hope these things are clear. These would help you in understanding in a better way how the mechanism of urine formation takes place inside the human body.